can have some, well, not so nice side effects. So basically, this is a heightened state of immune responsiveness to a foreign antigen, to an antigen of some sort. It can be contact, it can be respiratory, it can be food allergies, ingestion. There's a lot of different things it can be. There is some thought that it may be genetic because it does seem to run in families. Certain families, if your mom and dad had a lot of sinus problems and a lot of allergy, a lot of hay fever, a lot of times you see that with the children as well. So there's probably some sort of genetic link, but not necessarily. There's definitively proof now. They're talking about pregnant women should eat peanut butter, and one of the things that came out probably about a year, 2 years ago, a couple of good articles on maybe we're over cleaning around our children, that it doesn't hurt them to have a little mud in their life, that the immune system learns when you're an infant, when you're a child, when you're very little. And so if they are not exposed to a wide variety of allergens or a wide variety of antigens, it may be that their immune system, when it then is exposed, runs amok. It's just there have been several articles on it. There's really no 100% way of saying why you're going to be allergic to something or not, because there are children who are exposed to all kinds of things who develop allergies, and children who are in a practically sterile environment who never do. So there's a wide variety of things that actually contribute to this, and no one really has a definitive answer on why. There are basically 4 types, very creatively named 1, 2, 3, and 4. They use Roman numerals, but 1, 2, and 3 are an immediate reaction. You're exposed to it, you have a problem. The symptoms will develop at the most within just a couple hours after you've been exposed. With some of the severe, especially some of the contact allergies, you can have symptoms within just minutes. Hives, itching, rash, all the way up to anaphylactic reactions within just minutes. Delayed hypersensitivity. These symptoms develop a couple of days after contact with the allergen. These are usually IgE antibody mediated, and this one is usually T cell mediated. T cells are a little slower, it takes them a while.
You said the second one? 4 was what? IgM?
The first 3 are more B cell and IgE mediated, because we all know IgE is our allergy. IgE and eosinophils make you sneeze. Okay. Type 4 is more of a delayed hypersensitivity reaction. It's more T cell based, and none of these are strictly, but it's more T cell based, and that takes a little bit longer for it to come up. Type 1 hypersensitivity, this is what you think of most of the time as allergies, most common. It is IgE mediated, and what happens is these IgE molecules are sensitized, and they bind to specific receptors on the basophils and the mast cells, causing them to release what?
Histamine.
Histamines. And histamines do all kinds of lovely things. Okay. The allergens will cross-link 2 IgE molecules. Okay. So I've got these IgE molecules that are bound to the mast cells, or to our basophilic cells. And then the antigen connects 2 of those that are hooked onto the mast cell. The basophil, or the mast cell, then bursts open, and it releases histamine and heparin. Yes.
Question. And then this is over again. What's the circles at the mast cell?
Sure. All right, this is our mast cell.
Okay.
Okay, and these are those basophilic granules
Okay.
that we have in our mast cell. On the surface of the mast cell, we have antibodies that are bound to it. The constant portion of this IgE antibody binds to the surface of the mast cell, and it's sort of floating around. What happens is we'll get a cross-link between IgE molecules that are on the surface, okay? So this antigen will bind across these two molecules. And basically, it sort of pries open the mast cell, which then releases the granules that contain histamines and heparin.
Okay.
And heparin is the vasodilator, right?
Yeah, it is a vasodilator. Type 1 hypersensitivity, like I said, this is what you really think of as when you think allergies. These are the ones you think of. It can be inhaled, which will lead to tuffy noses and runny noses and watery eyes and all those, Claritin clear symptoms that we come up with. It can lead to trouble breathing. Asthma occurs basically if the allergen reaches the lower respiratory tract. Okay, so asthma is an allergy-related disease, although not strictly. Not everybody who has allergies has asthma, but people who have asthma, what happens here is that instead of it just sort of being kind of a rhinitis problem, being just up in your nose and making you all toughy and miserable, it actually makes it down into the bronchioles and down into the alveoli, and when these histamines break loose, it causes all kinds of problems down there, and it actually winds up basically squeezing so that you can't breathe. Your lungs are unable to get in enough air. Ingested, if the allergen is ingested, this would be your food allergies, or it could be, you could have an allergic reaction to something that you're not supposed to eat that you do anyway. Cow's milk, and would be an allergy to cow's milk, not just lactose intolerant. Lactose intolerant means you cannot process the lactose sugar from milk. So an allergy to milk and lactose intolerant are not the same thing. They do sometimes occur together, but they are two different things. Lactose intolerant means I cannot process lactose to get energy from it. And allergy means that the hypersensitivity pathways are activated. Okay. The most common really are milk, eggs, and seafood. I don't know why peanuts has become such a big problem. I really don't. If the allergen is injected, this would be things like insect venom, like bug bites, spider bites, even mosquito bites. A lot of people are allergic to the saliva of the mosquito, and they'll get a much worse reaction to it. This one is more likely, any kind of injected, is much more likely to lead to rapid anaphylaxis, although any of these can lead to anaphylaxis, which means your throat swells shut, you can't breathe, your tongue swells, pretty much everything swells, you stop breathing, and that usually is pretty bad for your health. It's usually pretty good for you. It can lead to shock. Basically what we're going to treat it with is antihistamines. If we get as far as anaphylaxis to where you're not breathing, where everything is swelling up, what they'll do is they'll give you a lovely shot of Epi. Yes.
Do bees, when they sting you, have some sort of venom?
Yes. They have a venom in them. It's not a super strong one, not like a snake venom or something like
Yeah.
that, but it is.
Like a mosquito?
Yeah, more like a mosquito-ish, yeah. The mosquitoes don't really have venom, they just have saliva that gets there, but, yeah, a bee has a mild venom,
And that's what the people are allergic to?
and that's
Not
what the
actual
people, yeah.
bee.
Not the actual bee, but the venom. So you shoot it with epinephrine.
And epinephrine is not an antihistamine, is it? Is it just
No.
a dilator?
It's a dilator and
And
I don't remember the other thing it does.
so,
It basically shuts down the immune response. But yeah, it counters the histamines. we test for this, well, we can do a skin test, where basically we're going to inject the allergen under the skin, and we're gonna see if it gets all itchy and yucky. And they'll do these multiples, and to any of you ever had it done or seen it, you've had it done?
I've had it done, my daughter had it done.
It's, from what I understand, it's not real pleasant, kinda uncomfy. But, a lot of times they'll have a little thing looks like a spider that'll do 8 or 10 or 15 of them at a time, or a little pad that has little like scratchy things with the allergen coated on it. So they just kind of lay it on the back and just kind of rub it on, and that injects the sites. And then they just come back and look at it, see.
It is painful.
Is it
20
painful?
minutes. They had me sit for 20 minutes. And then while they was they dig real tight up in your skin to try to get the allergen in your skin to see if you're allergic to it.
Yeah, they have to get it down into the lower levels of the skin. They can't just. We can do a RIST level, which measures just your total IgE. Okay, it's a simple EIA method. It can be done on a number of analyzers, but we can measure what your total IgE level is, how much you have. If you have elevated levels of IgE, obviously you've got some sort of allergic thing going on. The more common one that we do now, our IgE level actually goes along with our RAST panel. I'm trying to remember what they used to call them. They had another name.
Food allergen RAS panel.
A RAST panel is an allergy panel. You can have a food allergy RAST panel. We're actually Region 8 RAST panel. So if they ask for a Region 8 panel, we are Region 8. And what that means is, why would I test somebody in the Midwest for allergies to hibiscus? We just don't have a lot of hibiscus. I mean, there may be a few around, but that's just not a real common thing here. Why would I test somebody in Hawaii for ragweed? They don't have ragweed. So you want to test for the types of things that are around. You know, there's no need to test somebody in Alaska for I don't know, oak trees. Okay, but there's definitive reason to test everyone pretty much everywhere except Hawaii for allergies to pine, because that tree is kind of everywhere. So they pick the most common allergens and they start there. I don't remember how many. I think we're up to 18 in a standard RAST panel right now. They'll also, you can do a food allergen, you can do a pediatric food allergen, which they look at different things, fish, seafood, just a whole variety of allergens. Yeah.
Would you consider the skin test like a screening and the RAST like a confirming type thing, or?
Anymore, they pretty much just go to the RAST. I think simply because people don't want to go through
Oh yeah, it seems less invasive.
yeah, it is less invasive, less hands-on, quicker. Yeah, I'm not an allergist, so I really don't know which way they normally go with it. It may be your symptoms. Do any of you have had it done? Do you know why they do the skin rather than the blood?
They did my blood, both of them.
They did both.
They did both of me, cause I kept getting hives for at least 4 or 5 months while working at the restaurant, and I couldn't figure out what I was allergic to.
I said, I don't know what the criteria for why they do what testing. Type 2 hypersensitivity, kind of a two-step thing. It's going to require a little bit of assistance from either IgG or IgM. Okay, so this one is not IgE modulated. So what we have is our IgG or IgM molecules bind onto our red cells, which they are not supposed to do, but they do anyway. Then what happens is, because we have this antigen-antibody complex, it stimulates the neutrophils and the phagocytic cells to eat our red cells. This is a bad thing. Oh, isn't that cute? So it comes in and it eats it. this may also activate the complement pathway. And which pathway would it activate? The classical one, because we have our antigen antibody complex, okay? So the antigen antibody complex may also activate complement, which destroys the red cells. So when we get into blood bank, we're going to be looking for complement coating the red cells, because it is destroying them via a different method than the other. Types of hypersensitivity reactions. Really, these are pretty much going to be exposure to foreign blood cells. And pretty much the only way that's going to happen is a transfusion reaction or hemolytic disease of the newborn, okay, via across the placenta. If it's hemolytic disease of the newborn, it has to be IgG. Why? Because that's the one that crosses the placenta. You were listening. In a transfusion reaction, we receive incompatible blood. Intravascular hemolysis may occur within minutes, or it may be delayed for a couple of hours. Just kind of depends on how strong the antigen is, how rapid your antibody response is, whether you've been exposed to it before, and that kind of thing. And yes, you will have to know examples of each of these types of hypersensitivity. Basically, we've already talked about this. What's the difference between a direct antiglobulin and an indirect antiglobulin test?
Direct doesn't have a wash state.
Right.
Vivo and vitro.
In vivo and in vitro, which one's which way?
Vivo is the direct, in vitro, vitro is indirect.
Right. Here, we're looking for something that happened in the body. The antibodies coated these cells before we pulled them out of the body. Here, we're using reagent cells and taking the patient's plasma and purposely trying to coat those reagent cells with any antibodies that might be present in the patient's plasma. We do that in vitro. Type 3, this is an immune complex formation. We have our tissue cells here. The antigen combines with either IgG or IgM, and then it deposits, this complex will deposit down in the tissues, okay? So it sticks in there, and they're held in place. Then the complement, because we have damaged the tissue cells, and released all kinds of lovely things, is going to go ahead and activate the complement cascade. This complex may, by the classical, but frequently this will utilize the alternative pathway. And then the complement is going to come down, and it's going to attach to that antigen antibody complex, and those tissues are going to lyse. Okay, they're going to be, they're sacrificing themselves for the greater good. They're infected, so therefore they're going to be destroyed. Examples here, these are autoimmune type diseases. Okay, this is where systemic lupus erythematosus. Our antibodies against DNA and histones, these immune complexes deposit in multiple organs. This one says especially the kidney, but it can be anywhere. Okay, skin, kidney, liver, stomach, intestine. Rheumatoid arthritis, we have rheumatoid factor, which is an IgM molecule directed against IgG. So basically, you've got this anti-IgG IgM molecule that's floating around your body. It finds IgG anywhere and is going to glom onto it, and that complex is going to deposit, and for some reason it seems to deposit in the joints. that's where it kind of seems to live. Testing for this, pretty much we're gonna be testing, just like we talked about yesterday. We're gonna do the RF test. And how did we do the RF test? Latex agglutination. Can also be done by EIA. Do you have a question? Oh, no, just kidding, okay. Just try to make sure I'm catching. Or you can also do the ANA, which is the indirect fluorescent test. Okay, and that's the one, remember, that had the nice pretty little green pictures? Okay. Anybody remember which pattern we're looking for with lupus?
I know it's the one with no nucleus, probably. Nope. The one with
These
the
are all
scattered-
nuclear. These are all nuclear, anti-nuclear antibodies, so the nucleus is gonna be involved in all of them.
Is that the one where it's all green?
Right.
Yeah, that's the
Yeah.
one I'm talking about.
Oh,
with
that's-
no-
yes.
Yeah.
Yeah, no little
It's
dots, yeah, okay, I'm
yeah.
sorry, I
I'm
misunderstood
sorry.
what you were
okay.
saying. I was thinking in reverse. But yeah, the nucleus is positive, the whole thing, that's the homogeneous pattern. Okay. Anybody remember what we go to if we think it is lupus? What test do we go to? Instead of an anti-ANA, instead of an ANA, what do we go to?
Anti-CCP?
Nope.
Is it the double strand?
Double-stranded DNA. Anybody remember the critter we use?
The cute one with the tail?
If you own the tail, Crithidia. Okay. Type 4 hypersensitivity is different. Okay, it's an entirely different pathway. There are no antibodies involved, and it does not involve complement. This is why this one takes a little bit longer to do. Okay. Basically, this is going to be a T-cell mediated hypersensitivity. We're going to have some sort of stimulus, bacteria, a virus, a fungi, poison ivy. This is how this one works. Anything that gives you kind of a not just the hives and the kind of itchy red skin, but anything that kind of gives you a bumpy, nasty rash kind of thing. This is also how PPD works. When we put in the protein purified derivative, that's what PPD stands for. We put that in, and this is the pathway that we're looking at when that happens, which is why it takes a couple of days before you can read it. The CD4 cells are activated by antigens, and then they release the lymphokines. CD4 finds these antigens and says, everybody come help me. Neutrophils are then brought to the area by these lymphokines, and this leads to an inflammatory reaction, okay, which is heat, pain, swelling, and redness. So the neutrophils come in for lunch, and then the cytotoxic CD8 cells come in and destroy the antigens. Types of type 4 hypersensitivity: contact dermatitis, things like poison ivy. Can anybody think of anything else? Nothing's coming to mind here. But anything that like you rub up against and it gives you a rash.
Pollen.
I'm sorry?
I said poison.
Poison sumac or poison oak, yeah.
Pollen.
Pollen, not so much, that's more an immediate, it's just hives, it's not really a long-term.
Like my brother's thistles and grass.
Yeah.
That's in hot
Okay.
tub, that helped right that.
Okay.
Does it matter on the person? Because they say sometimes, like, some people have contact dermatitis, they're allergic to bananas if they touch a
Well,
banana.
and that could be
So
too.
does it matter?
Yeah. But the key here is that it gives you not just hives, you know, because hives go down pretty quick. They're supposed to go down pretty quick. Alright, but they're kind of immediate come up. But poison ivy rashes or things like this, they're harder, they're thicker. I don't know how else to describe it, but they're more of a granulomatous type rash. Hypersensitivity pneumonitis. What happens here? This is basically you get a rash on your lungs. Okay, so what you wind up with is these sensitized T cells responding to these inhaled allergens, so this reaction's occurring in the lungs is not a good thing. Granulomatous hypersensitivity, most commonly this is TB. Okay, so testing for exposure to TB is a type 4 hypersensitivity reaction. That's the PPD.
The granulomatous, what is that standing for?
Granulomatous, well, basically what it is it walls it off. The T cells all kind of come in and they find it. They bring in the neutrophils, and it almost makes like a wall around it. So it makes a plaque or a hard spot where it surrounds it and kind of keeps it from spreading anywhere else in the body. All right, questions? All right, those are the immune system disease states. We still got quite a bit of time. Why don't we go ahead and move on to spirochetes? Just take a minute, print it for those of you who need it printed. Or if you want, we can do HIV because we're going to be doing an HIV test today. Would that make more sense? Let's do HIV. Everybody okay with that? If you don't have it printed, we'll print it. We'll take time. It's fine.
How many what?
I do.
On Monday.
I added to.
Yep. Yep.
Holy Mondays. I'm scared of it already. So many.
Well, yeah, but there won't be a lot of room, so it's still going to be a lot. I will narrow it down for you Friday. I will narrow it down for you Friday. And if it really looks like it's gonna be way, way too much, like I said, I may do a take home with the virals or something like that. Cause there's only 3 more, and really these aren't that horrendously long. I mean, hypersensitivity was what, 6 slides? HIV is actually a relatively new disease state. First came to the U.S. Back in the 80s. So you have to remember that when I went through training, this was still that gay guy disease in San Francisco. It really was not, so I've kind of sort of, in my career, I've sort of grown up with it, so to speak. Some of the original assumptions about this thing were, I mean, they were like, what's causing it? They had no idea. They were getting all kinds of weird disease states, all kinds of odd stuff coming up. Basically, HIV is the human immunodeficiency virus. It is a cute little guy with a nice little lipid outer envelope. It comes in and it infects your cells from the inside. And the problem is the cells that it does infect. HIV-1 is the most common cause. There are 3 distinct serologic types. There are infections worldwide and in West Central Africa. It was first found in 1981. And in the U.S., the majority of cases of HIV are HIV-1. I've never seen an HIV-2. I don't think there's really more than a handful of them. HIV-2 comes more from West Africa. Transmissions is the same. It's less pathogenic, and that may be why we don't see as much of it. It's also less easy to transmit. So basically, if you've got HIV too, you're less likely to infect somebody else, and it doesn't cause quite as bad of a disease state. So we don't see it nearly as much as we do the other. They are also called HTLV1 for human T lymphocyte virus antigens 1 and 2, or I'm sorry, lymphotropic viruses. And if you do remember back to hematology, no, don't make us remember. Back to hematology. What else is associated with this virus? Do we have some lymphoproliferative diseases that were AIDS, that are induced by AIDS? Smile and nod for me. Couple of lymphomas, if you will remember correctly. And even if you don't remember, there were several lymphomas. It is a retrovirus. It is an RNA virus. Okay. The RNA and an enzyme called reverse transcriptase transcribes the viral RNA, so it goes into your cell, and the viral RNA is then translated to DNA, which pretty much takes over the cell and says, stop doing what you were doing and make more of me. It is spherical. It's actually fairly large for a viral particle. The outer envelope is a glycoprotein with lipids, and this binds the virus to the host cell during infection. Okay, so it actually kind of holds on to the host cell and allows the virus to be injected into the cell.
So they're hijackers.
Pretty much. There are 3 main genes in HIV: GAG, ENV, and POL. These code for the glycoproteins on the outer membrane. Okay. These are in both HIV-1 and HIV-2. So when we're looking for testing on the HIV, a lot of times we're looking for antibodies to those glycoproteins. GP 160, 120, and 41. And kind of the biggest one that is important for blood banking is this P24. Initially, the tests that came out were antibody testing only. So you had to look for the antibodies in the donor blood. And if the donor didn't have a high enough titer of antibodies, you know, you're going to get somebody, they're going to get slipped through. They developed this P24 test, which means that they go and they look directly for the virus and scan directly for the virus. So that hopefully those patients who didn't seroconvert, they'll still catch it. The important thing here is that it affects the CD4 cell, and which are our CD4 cells? Those are our helper cells, okay? It infects the T helper cells so that they can't help anybody. They're too busy fighting off the infection. They've been hijacked so that they are making more HIV virus instead of doing their job, which is stimulating the immune system to make memory cells and cytotoxic T cells and antibody production. So the HIV molecule attaches to the CD4, and basically then once it attaches here to the surface, the membrane at the backside kind of opens up, and the viral particle then goes into our CD4 cell. Viral replication occurs and new virus particles pop out of the host cell, which lyses our CD4 cells, so it blows them up. This is how most viruses work, whether it's a rhinovirus or what kind of virus it is. The reason this one's particularly bad is that it's affecting our immune system, which is supposed to fight this stuff off. So the viral particles then burst out of the host cell, and then what do?
Go to other T cells?
They find more CD4 cells and attach to the surface, inject themselves in, and replication occurs. Transmission is through sexual contact, both heterosexual and homosexual. Contaminated blood or body fluid, a shared needle with IV drug users. Another one that was a big stink back in the early 80s people would sell their plasma. Okay, and a lot of the people who would sell their plasma were IV drug users, because, you know, that's a good way to get money for drugs. Unfortunately, what happened is the HIV got into the plasma supply, and it doesn't take very much of it, because they would pool hundreds or thousands of units of plasma together and then pull out the components they wanted, most notably anti-hemophiliac factor, which is what? Factor's what? 9, and more commonly, hemophilia A.
That's 7, isn't it?
Nope. Hemophilia A.
No, I'm thinking Hep.
What factor?
13?
Keep going. You still got half a dozen left.
Hey!
I've been doing this to them all semester, making them remember things. I mean, factor 8. A is 8. A is
Hep A.
8.
Hep A and Hep B, I thought. That's Hem A and
I'm sorry, what? We're talking hemophilia A and hemophilia B.
A. And you're talking about the clotting factors
The
from...
clotting factors, and
Okay,
yeah.
hematology. But I thought... Okay, I'll have to go look at my... Yeah.
So what happened was they collected all of this plasma from, and this was purchased. This was not the, Red Cross donor supply at that point. This is people bought plasma. And so what happened, unfortunately, the factor 8 supply for hemophiliacs was contaminated. And well, when you've got hundreds or thousands of units of plasma being, basically boiled down for the factor 8, and then lyophilized, many people who were hemophiliacs wound up with HIV from their anti-hemophiliac factor. Most famous case was a young boy named Ryan White. I don't know how many of you were around for that. Any remember it? You'd have been
They made a
fairly young.
movie about him, didn't they? He was a swimmer. They made a movie about him. He was a swimmer. He banged his head against the...
No, he's a young kid. We learned about him in science
I
class.
say he was a kid, and there was uproar. They didn't want him to come to school, but like I said, this is back before they really knew how it. Remember, I told you they did studies on people who shared toothbrushes, or razors, or just lived in houses with people who had full-blown AIDS. They were trying to figure out how it spread. They really didn't know. Of course, you had those who said God was striking the evildoers, but well, doesn't usually work quite that direct action. But until they really figured out what was going on, the initial population that was most affected were gay men. So it took them a while to figure it out. Once we learned how to screen for it, the anti-hemophiliac factor became safe again. Congenital, it can be passed from the mother to the fetus, either during pregnancy, delivery, and it can come through the breast milk. So, that's why we say when you're looking at, who is infectious or who is possibly an infectious person, you can't rule anybody out, whether it is a newborn baby, because they could be infected with anything mom had, or a 95-year-old lady. Yeah.
I want to ask a question. Okay,
Sure.
are they doing these tests before they have the baby or after? They give them syphilis, HIV, and
Once they
chlamydia,
get pregnant, they do the screening tests.
not like during pregnancy or nothing.
During
like that?
pregnancy,
like when
yeah.
Like when they're about to have the baby or something like that.
If they haven't had prenatal care, includes doing those screening tests.
Hmm.
If they have not had prenatal care when they come in to deliver, the test will be done at that point. But usually they're done in your first couple of doctor's appointments. And they'll also do a type-in screen at that time to look for abnormal antibodies.
They have breast milk supplies now.
Mm-hmm.
Are they screened
I have
the
no
same as
idea.
blood is? Because people donate it to just give out to others that are having problems. And that's not good.
I don't know.
Are they donating it through a hospital or something? Because that
No,
sounds like
no,
a...
No,
these are like
it's-
private.
There's
Probably not.
not-for-profit organization probably, but yeah, no, I have no idea. I'm not, yeah, I'm not a, I don't know. I don't know if it's regulated, I don't know who would regulate it if it is. Yeah, I don't know how that
They sell it on
works.
Craigslist.
Yeah, you can.
Yeah, oh, I'm sure they sell everything on Craigslist.
Websites.
Yeah.
I've never seen people buying it, but I have a friend who's donated and, not a friend, an acquaintance, she's donated and she's like accepted it from other people.
Too close.
Yeah, I doubt it is screened. I would really, really doubt, that there's any kind of screening process on it, because it's natural, and, you know, we don't need to screen natural things. I don't know. I really don't. I don't have enough information on the process.
I'm sure it will be when somebody's baby gets.
What is, yeah, when something goes wrong and somebody gets sick, then they'll swoop in, but.
The HIV kits that you can buy over the counter, would you be able to screen using
I don't-
those?
It would depend on the kid. If it's probably not, because most of them are screening for antibody. But you'd have to look at the kid, for sure. The HIV infection cycle, it starts with, basically, it's kind of flu. You just don't feel real good, get swollen glands, fever, fatigue, you know, kind of a mononucleosis type infection. You just don't feel good. It really doesn't necessarily last very long. It's usually about 3 to 6 weeks after the infection. It takes a couple weeks before it starts to really cause a problem. But basically at this point, the virus is kept in check by the immune system. Your immune system is able to overcome the virus infection at this point. It's still adequate. Then you wind up with the latent period. And this can last for a very long time. I don't know what the latest figures are. This says up to 10 years. I do know that I've heard anecdotal that there are some people who haven't seroconverted even after 20 years, that they test positive for the antigen but still are antibody negative. They are considered HIV positive at this point, but if you do the basic HIV screen, you know, where they go into the ER or the Planned Parenthood or your local clinic or wherever they're going for testing, they'll show up as negative because they don't have any antibody. So if we're testing for the antibody, they're going to be negative. But they are HIV positive, it's just that they don't have the antibodies going around.
Is that why you get a non-reactive instead of a negative?
Yes, that's why we report them as non-reactive instead of negative. Simply because if they are in this latent period, there's no way for our test to tell if we're testing for an antibody. Yes.
If you know that they could be positive but still not be reactive, why would they even have that testing still instead of just testing for the antigen?
Time, money, costs a lot more to test for the antigen. Plus, if you're in that latent period, you probably don't have enough viral particle to pop up necessarily. So no HIV screen is 100%. No any screen is 100%. That's why they're all sensitivity 99.99999. Why? Because then if you're that 1% that it didn't pick up, they can say, see, we said 99.9999. Okay, we didn't say 100%. AIDS is the active stage of the infection. This is when you get full-blown destruction of the immune system. The virus, for some reason, is reactivated. It could be your immune system becomes compromised for some reason, and it could just be you aged, you got sick, something else is going to go wrong, and basically what's going to happen is the virus is no longer going to be contained, and you're going to have active destruction and infection of your CD4. Your CD4 counts are going to drop, okay? an absolute count of less than 0.2. So the patient is going to be susceptible to opportunistic infections and malignancies. The symptoms are going to include things like night sweats, skin lesions, neurological problems. When I was going through training, like I said, I sort of grew up with AIDS. I cannot tell you the number of times, especially in things like mycology and those departments, oh, you never see these diseases. Except in those Dr. Neibauer patients, and Dr. Neibauer is the guy who treated all of our HIV patients. The things that most people, you never saw, you know, histoplasmosis. You don't see histoplasmosis once in a great blue moon because it's a fungal or a fungal infection. And your body just doesn't usually have systemic fungal infections. I mean, you get athlete's foot or ringworm and things like that, but you just rarely have systemic fungal infections. The CD4 cells and the immune system itself, pretty good at recognizing those things and blowing them up before they get to be infective. Well, we started seeing histoplasma, aspergillus, all kinds of weird things. And like I said, the lady who taught microbiology, she probably said it 1,000 times. We never see this. Well, you know, except all those Dr. Neibauer patients.
Where does the virus live?
Inside the CD4 cells.
Even when it's not activated? So why can't we transfuse people? Why can't you
Replace the CD4 cells. What is the main purpose of the CD4 cells? What do they go around looking at?
the MHC?
The antigen.
Right, they're looking for foreign invaders. Where am I going to get new CD4 cells from? Because if I get them from somebody else,
Oh, yeah, it's not gonna work.
right, not gonna work. Yes.
Does behavioral problems fall under neurological problems? Because I've noticed like not HIV, but every AIDS patient we have, they have to have like sitters and
It's basically, it's affecting the brain. The neurologic problems are not, it affects the brain and it affects the neurons up there, which can lead to mental problems. I don't know the exact mechanism on it, but yeah, I do know that they say that psychotic episode, well, and even some of the infectious agents that they get can wind up infecting the brain.
Okay.
And I, without knowing specifics of the cases, I can't be more.
Right.
But I do know that as they have more and more neurologic problems, it does affect the neurons in the brain. How or to what extent, I really don't know.
So, the CD4 is being affected by the HIV.
Virus.
It's not directly affecting the brain, it's affecting something that's keeping the CD4.
Well, the CD4 isn't able to protect against the things it normally protects against.
Okay, so it's not the HIV that's affecting the brain,
Yeah,
it's
the
something else
HIV
that's
doesn't
being...
directly, it doesn't directly attack anything but the CD4 cells. The problem is, because the HIV is attacking the CD4 cells, it is decreasing your immune system to the point where you can't fight off the other things. And those then come in and cause the damage.
So
So
CD4
HIV doesn't directly kill you. It's not like it comes in and, explodes your lungs, but it comes in and destroys your immune system so that other opportunistic things can come in and kill you.
Okay, but the CD4s, they're the ones that recognize antigens. They don't recognize HIV as an antigen? And that's why they
Well,
allow
they
to touch
do,
it?
but the HIV is able to sneak past.
Okay, because lipid...
Well, you make antibodies, don't you? We're testing for HIV antibodies. So yes, you do make antibodies against HIV, but what happens is the HIV manages to get past your immune response.
Do
And
you know
it
why
doesn't
that
take
is?
much of it.
Do you know why that happens or how that happens?
Yeah, it goes into the CD4 cell.
Yeah, but usually the CD4 cell is saying, hey, antigen, let's latch onto that. And it's latching onto it itself, so why is it just stopping there?
Well, it doesn't. It hijacks the DNA in your CD4 cell. So instead of your CD4 cell looking for antigens, your CD4 cell is busy making more HIV virus particles. So basically what happens is that you just get enough HIV that that it infects your CD4 cells to a point where your immune system can't stop it.
I get that, but the CD4 is what's supposed to be recognizing the HIV.
Yep.
And stopping it before it ever touches it.
Yep.
How is it hiding from the CD cell
It's
before
sneaky.
it's okay?
It's getting to the membrane, and because the CD4 is looking for it on the MHC. Remember, the CD4 looks for the antigens on the MHC molecules.
On the antigen presenting cells. So it's not going to see
Right.
it if it's just free floating in the blood.
Probably not.
Okay.
The opportunistic infections, and this is, this one just is a few. Candidiasis, what is candidiasis? Anybody got a clue here?
Something swollen?
What's candida? It's a yeast.
Yeah.
Candida albicans. It's actually part of the normal gut flora, a little bit is the gut, but it is definitely a normal portion of female vaginal flora as well. And it's found all over the place, it's pretty ubiquitous. It is most commonly causes vaginal yeast infections and thrush. Okay, but what happens here is, vaginal yeast infections and thrush, those are pretty much external. Okay, those are not systemic, those aren't within the body. Okay, candidiasis in this case can infect the brain, the liver, the muscles, the bone marrow, places it really shouldn't be. It's not just a surface type of infection. Kaposi sarcoma, and yes.
I have a question back to the last slide, I guess.
Mm-hmm.
Why couldn't you treat with chemo?
They tried, it didn't work.
I was like, 'cause
It was more damaging. It's one of those things, chemo, most of chemo is a poison. And you didn't knock out all of the HIV cells, so it just came back.
Doesn't it suppress your immune system?
It suppresses your immune
And
system.
you're already suppressed being HIV positive or
Well,
AIDS?
they tried, they thought, well, maybe if we can, do an anti-CD4. And I've only read one trial on it, so I don't know how hard they tried or what they tried or exactly what the agent they used was, but chemo did more harm than good.
If you do a bone marrow transplant, can you completely reset your blood system? Your supply of, because you said you can change your blood type.
But the lymph cells aren't just in the bone marrow. They're elsewhere as well. So if you do a bone marrow transplant, it'll get some of them, and it gets most of them. But but again, then the virus is still, you're not gonna get 100% of them. So if you get new ones in there, that's great.
They'll just
But
get infected again.
They'll just get infected again, yeah. Believe me, you guys are really smart, because they've tried most of this stuff. Sounds logical. Yeah, and like I said, I bone marrow transplant is not one that I think they've tried, but it does make sense why you would. But again, then, when you have a bone marrow transplant, what do you have to give them? Anytime you have a transplant, what kind of medications do you have to give them? Immunosuppressive therapy. Not gonna work. Not gonna work. It's a slippery little dude. It really is. The best way with most of these viral, but the best way to handle most of these is epidemiology, is to keep it from spreading. Kaposi sarcoma, any of you see the movie Philadelphia?
Yeah.
Okay, this is how they actually started putting together all the little pieces and parts. This is a cancer, it's a little lesion that pops up on the skin in various assorted places. Again, it's one of those things you saw once in a billion, people, and suddenly all of these people are popping up, I mean, lots of people are popping up with this Kaposi sarcoma. So this really, really rare disease is suddenly affecting large numbers, relatively large numbers. So they were actually able to start following it, and then as they followed that, they were able to kind of branch off and go, okay, look, we can see here, and they were able to kind of narrow it down. And they narrowed it down to basically one flight attendant from France was responsible for a large portion of this. But they were able to kind of take it back and figure out where it came into the country at. And it came in up in San Francisco area. So that's kind of where it started. But they were able to then follow it, and as they were following, they followed the Kaposi sarcoma, but then they noticed other trends amongst the people that were having this, and so they were able to, over many years, sort of follow this and figure out what was going on. Burkitt's lymphoma, remember I told you one of the lymphomas, that's our HTLV1. And does anybody remember what those look like? Those are the weird ones. How do we tell Burkitt's cells?
Those the dumbbell looking ones.
The pince nez.
The what?
The pince nez.
No.
No, that's Poker Hewitt.
Those are neutrophils. They're the ones that look like somebody took a hole punch to it. Okay. You would see pneumocystis pneumonia. Again, this is another pneumonia that you just don't see. It's not common. For the most part, our body is able to, if it does get in, our body is able to wall it off. It's able to hide it, or to sort of segregate it, to put it away until it dies, or at least doesn't spread. But with the decreased abilities of the immune system, it's able to take over. Toxoplasmosis. This is one case where you would definitively have brain problems, because when you have something eating away at your brain, it's not good. But you will get infections of the brain, which brain infections aren't real common. Encephalitis and meningitis, some, but for the most part, your body protects its brain pretty heavily. A lot of other opportunistic infections, viral, parasitic, tumors, okay? Anything that needs a T cell to help fight it off, without that CD4 cell to basically tell it what to do, it doesn't know to fight it. And so it pretty much just breaks your entire immune system down. So this is, it's not the common illnesses, it's not strep throat, it's not, the things you see a lot of. It's the weird stuff that the T cells fight off. Your fungus, your parasites, your tumors. Okay? Yeah.
Does the virus change the MHC complex on the outside of that cell? Or would that even matter?
No, because there you're looking at class 2 that's presenting antigens, right? And the viral infected, that's a good question. What happens to the MHC? No, because you would be looking at the MHC1 because it's a virally infected cell. So what does it do to the MHC1 receptors on an infected cell?
Or does it do nothing?
I'll have to get back to you on that one. That one's not striking up right now.
So is there like research that goes on for this the same as cancer research?
Oh yeah. And 80s and 90s, hundreds of millions of dollars were poured into trying to find out what was going on. Because at that time, it was 100% fatal, 100% of the time. I don't remember how long Ryan White lived. I want to say it was less than 5 or 10 years. He died fairly shortly after all the stuff happened. But yeah, it was 100% fatal. It still is 100% fatal. We just can stave it off a little longer. Once you get to the AIDS stage, it's treat symptomatic. It's treat the diseases as they come up. What the what the antiretroviral drugs do is to try and keep you from getting from the infected stage and to keep you from going from latent to HIV, to full-blown AIDS. Basically, you're gonna do supportive care of the infections and the malignancies. You're going to try and fight them off as you would any other patient. There are several classes of antiretroviral drugs being used. A lot of them are reverse transcriptase inhibitors. The key one to start with was AZT. Unfortunately, viruses are extremely adaptable little suckers, and AZT is becoming less and less effective. That's why if you hear about somebody who has AIDS and is being treated for it, a lot of times they'll get what they call antiviral cocktail. And they pretty much have to keep adjusting that cocktail because the virus keeps mutating to adapt around what antivirals are being given to them. So a lot of times they'll give them these antiviral cocktails. They may work for 6 months, a year, 5 years. You know, it just depends on, person to person. But then it stops working and you've got to kind of remix the cocktail and try something different. And these drugs there's less of these than there are antibiotics. So it's not like, okay, we're going to start with penicillin and then we're going to move on. And we've got tons and tons and tons of choices. These antiretrovirals, there's not nearly as many of the different choices. They're making more, they're getting better, they're learning fast. But the problem is, once it becomes resistant to that particular one or that particular cocktail, it gets harder and harder. Another problem with treating this is that you have to take these things on time. It's not like, oh, my blood pressure medicine. Oh yeah, I forgot to take it. Well, I'll get it in a little bit, it'll be fine. My antibiotics, I take it in the morning, I take it at night. If I don't take it at 0800 and 2000, I'm okay. These little buggers gotta be taken religiously. Like, the first sets of people who were on this are given watches with a timer. And when the timer goes off, I don't care who you are, where you are, or what you're doing, you will stop and you will take your medications. Because if they're not taken exactly according to timing, they stop working. So, if you miss a dose, it's very possible that it will overwhelm your treatment. So, they're very difficult, and they are horrendously expensive, thousands of dollars a month for these medications. Multi-drug regimens are used to treat the opportunistic infections and block viral replication. Highly active retroviral therapy or HAART therapy, again, is another combination of drugs to kill the virus. This seems to be more, it was one of the cocktails that seemed to work a little better, or a class of cocktail, I'm not sure. It's more of a class of cocktails. Like I said, they kind of are playing with the different antiviral drugs to try and find one that will block the reverse transcriptase effectively. Because if you block that reverse transcriptase, the RNA that goes into the CD4 cell can't be turned into the DNA. And if it's not turned into the DNA in the CD4 cell, the CD4 cell doesn't make more virus. And if it doesn't make more virus, when it blows up, it doesn't infect other cells. Okay, so by blocking the reverse transcriptase, you're blocking the ability of that virus to replicate and for it to infect other cells.
But you're also blocking the other healthy T4 cells to
Sure
replicate
are.
as well.
Yeah. What kind of testing do we do for this? Well, one of the things we're going to do is look at the CD4 absolute counts and ratios. And most commonly, the most important thing we're looking at is our CD4 CD what ratio?
2.
Nope.
Oh, 8.
8.
8.
CD4 CD8 ratio. That's the most common ratio we're looking at when we're measuring. We do an absolute CD4 count, but we also watch the CD4 CD8 ratio. If the CD8 starts to increase too much, and our CD4s start to drop, we start having a lot of problems. How do we measure those?
Flow cell cytometry.
Flow cell cytometry. Yeah, flow cell cytometry. As a matter of fact, there are quite a few analyzers out there now that can do a CD4, CD8 count just in a basic lab. Doesn't have to go to a special flow cytometry lab. That they have something that can do it. Now, the thing there is you have to do enough of them to make it worth putting the reagent on, doing all of the QC, doing all of the calibrations, and maintaining all of the extra records you have to do for that. So a lot of people, even though their analyzer will do the CD counts, they don't use it simply because it's just more hassle than it's worth. It's cheaper to send it to somebody else. We can do antibody detection, and we can do antigen detection. Most of your HIV screens are going to be antibody detection, and that's what we're going to do today, our little Edvotech HIV test. We're going to do a manual EIA test, okay? Instead of just putting the stuff onto the little kit and watching it turn colors, we're actually going to add each little piece into the little well and see what we're doing and see it step by step by step how we do the test. We can do antigen testing. We can test for the viral antigen nucleic acid or for the nucleic acids itself using a PCR method, using a molecular diagnostic method. Okay. But again, PCR is expensive, complicated. So a lot of times when we're doing this stuff, we have to have a screening test, and in a screening test, I'm looking for something with high sensitivity. I'm looking for something cheap, easy to do. When I'm doing a lot of our confirmation tests, then PCR becomes our confirmation for a lot of this stuff. What is the confirmation for HIV though? We talked about the Western blot. The Western blot. And you have to have 3 of those antigens for it to be positive. So we can do a CD4, CD4 lymphopenia is the hallmark feature of AIDS. And there's a difference between HIV and AIDS. HIV is an infection with the HIV virus. AIDS is the full-blown expression of that. That is when you become symptomatic. That is when you start having these infections, these cancers and the other stuff. So that is, AIDS is the actual disease state that comes from HIV. We're going, for our CD4, we're going to do flow cell cytometry. We're going to report it as a absolute count. We're going to report it as a percent of the total lymphocytes, and frankly, if you're counting them, it's just as easy. They usually report it out all 3 ways. You know, this is your absolute CD4 count, it's this percentage of your lymphocytes, and your CD4, CD8 ratio is this. They're doing it anyway, it's not that hard to report them all out. In general, for an absolute count, if we see less than 200 per microliter, or less than 200 per times 10 to the third, times millimeter cubed, our patient is going to be diagnosed as having AIDS. Although there is usually a component at that point that says they have to be symptomatic as well. That caveat comes from the insurance companies, not from the medical community. Okay. That they won't call it AIDS unless you are actually having some sort of infection. Which, if you have AIDS, you will. If you don't have one right now, you'll get one pretty quick. Okay. We can do an EIA screening test. It's a non-specific test for anti-P24, anti-GP41, or 120 and 160. In other words, it's looking for antibodies against a variety of epitopes. Right? Each of these is a single epitope. When we're doing our screening test, it's non-specific. We're just looking for any antibody against any of the capsid or any of the protein markers or lipoprotein markers on the cell or on the surface of the HIV virus. Okay. Samples which are repeatedly reactive are sent for confirmation. So the first thing I'm going to do if I get a reactive first thing I'm going to do if I get a reactive HIV, The first thing I'm going to do is I'm going to repeat it. Some places actually make you draw another sample. Again, anytime a new disease comes out, people are very scared of it. Why? Well, it's scary. And this one was a particular nasty little monster. So, say back even in the 90s, early 2000s, the rules for reporting HIV were totally different from anything else, because supposedly it was worse than anything else. As it has become, as we've studied it longer, as it's been around longer, they've realized that it really shouldn't be reported any differently than any other of our STDs or any other of those disease states we monitor. Truthfully, this guy is a wuss. If it doesn't have a white cell to live in, it won't live very long on the counter. You wipe down the counter with a nice disinfectant, it's gone. Hepatitis? That's a persistent little bugger. Okay? It can be infective even without a cellular source around. It can live on the counter just quite happily, just sitting in the air doing nothing. And suddenly you rehydrate it and it's back in business. So it's a much more persistent, a much more difficult to kill virus as far as infection control is concerned. HIV, pretty wussy. I don't remember the numbers, but I think it's like 2 hours. Something like that. If you've got a blood spill on the counter, it can only live for a couple hours. Hepatitis, a couple days. So it's just a matter of epidemiology, protecting yourself. The Western blot tests for the same antibodies, but basically it tests for them individually. Okay, it's going to add an electrophoresis type step or a chromatography step that's going to separate out the different antibodies. So up here, we're just testing for a glob, any of these antibodies present. If there's 1 or 2 or 3 or all of them, we're going to get the same reaction. When we get down here, we're going to separate them out. And if you have to have 2 out of the 3 antibodies in order to make it a truly positive test. Does that make sense?
Is that an insurance thing again, or is that?
I think it's a little of both on that one. Okay. Just because the medication that you give an HIV patient is so damaging to the rest of their body, it's so expensive, and it is, if you treat them too soon, then it won't work when you actually do need it. So I think it was both a medical and an insurance. I'm sure driven mostly by insurance, but I think it was both. That if you've only got 1 of the antibodies, your body is still handling it well enough that we don't want to institute treatment. You start to see more of those antibodies. That's a guess on my part, but yeah, I do know it was both insurance and medically driven for that. The P24 antigen testing uses a solid phase antigen capture enzyme immunoassay. There's 2 antibodies, it uses 2 different monoclonal antibodies against P24. One coats the solid support, and one is added that's tagged with an enzyme. We do a confirmation, and we confirm, I'm gonna try that statement again. We confirm positive results using a neutralization procedure. We do the same thing for our hepatitis testing. If we get an antigen, an antibody to the antigen, then we neutralize the antibody and do it again and see what happens. This is a picture of a Western blot. Here's the P24, the GP1 2160. There's also a P32, 42. Oh, I didn't know there was a 51. Okay. So there are multiple antibodies, but you have to have antibodies to at least 2 of the 3. PCR, the other thing we use PCR for is not just to confirm or to find the viral antibody, Well, we can actually, and we use this in multiple viral infections, not just HIV. We also use it in HCV and some of the other nastier little viruses that are out there. We can actually count how much HIV is in your system. How many particles are actually in your system. And the way we do that is with PCR. Okay. And just like you talked about it in Barb's class, we're gonna, I know what I'm doing this weekend. We're gonna talk about it next week in here. We're gonna talk about a couple of the different methodologies you can use. True PCR, the biggest thing here is that it multiplies the DNA in order to measure it. Okay, it strips it open, makes a template, anneals the primers to it, extends it, and then you measure the products. The thing is, there's so little of it here that what it does is here in this annealing and extension phase, it will take and make millions upon millions of copies, so there's enough there that we can actually measure it. But it will tell us, it will then tell us how much of the virus is present. And they can do that when they're trying to treat it. They can use that to monitor treatment to see if it's doing any good. Are we decreasing the viral particle count? And we have some doctors who want to go straight to this for screening. The answer is no, for multiple reasons. Number one, it's very specific, but it's not a good screening test. It's good for somebody we know who has it, but if they have such a low particle load that we don't catch it, that's a bad thing. Okay, so
Quantitative instead of qualitative?
it is quantitative. This is a quantitative HIV. So if the doctor requests a quantitative HIV, and I had a person who does our orders lab tests, didn't order the quantitative HIV. She said, well, there is no such thing. First of all, never say there is no such thing, because they're coming up with new stuff, like, I think daily. It's just to confuse us, that's all it is. But there's new stuff all the time. But yeah, there is a quantitative. It's just not called quantitative in our test catalog. It's called HIV by something PCR. It's not straight PCR, but yeah. HIV DNA by PCR. So it's just called something different. Questions, problems? HIV's fun stuff, right? The thing to remember, it really is not that much different from any other virus. The biggest piece with the HIV is its target organ, because every virus has a target. What does rhinovirus target? Or node. It's what gives it a code. Okay, so a rhinovirus targets your nose. What does the hepatitis virus target?
Liver.
Your liver. Other viruses have other targets. Okay. The thing is, with HIV, it's targeting the very system that is designed to protect you from viruses. So unfortunately, what happens is it destroys the immune system. And as it destroys the immune system, your body becomes susceptible to all of the other things that your immune system protects you from. So that's why HIV is such a different, why it's kind of its own category. It's also its own category because it's relatively new. Okay, something that's only been around for 30 years is new. So it's something, and as we treat more and more diseases and we're able to overcome Mother Nature's attempts to get rid of us, we're gonna start seeing